This conference will now be recorded.
Hello students, I'm Dr. Divya from TeenEinstein. So here we are starting biology classes of 9th standard. The first chapter of biology, that is the fundamental unit of life, and this is part 1. So in this chapter, we are going to learn about what are living organisms made up of. Okay, so with what all these living organisms are made up of, you can see different types of living organisms, living things around you. So what are these living things are made up of? What that we need to learn. Then what is a cell made up of? Cell and cell components. About that we need to learn in this chapter. These are the main topics. Under this we'll learn each and everything very clearly. So let's start. The fundamental unit of life. See, actually when we are saying biology, which is dealing with the cell, that is called as cell biology. Okay, we are going to learn about cell biology, the biology of cell. Okay, so that is the branch of biological science which is dealing with the aspects of cell, about all aspects of cell. Like we can say cell biology deals with all aspects of cell. In the sense, we can say that maybe morphological features, physiological features, maybe the biochemical features, then genetical, then developmental aspects. About all these things we are going to learn in cell biology. So this in 9th standard, this is just like a starting point. You are going to learn about cell biology, the biology of cell. Okay, biology in the sense about all we are learning. Morphological, about the morphological features, external features, how the cell or how the morphology of the cell is. Then physiology of the cell, then the biochemical properties of the cell, then the genetics of cell, then the developmental aspects. Everything we are going to learn in cell biology, right? So here this is like a starting step. You're going to learn about the general things about all these. Okay, so we can say that it's like a new branch, and this gives a new dimension also. Like the birth of cell biology, birth of cell biology. How such a new branch formed? Like cell biology, plant biology is there, animal biology is there. So cell, when that concept came, like up to a cellular level, how we started about those things? It is through the invention of compound microscope. Okay, invention of compound microscope, right? See, we are seeing our hands, legs, everything, right? But it is starting from the cellular level. Or if you are cutting a plant, we can say, or cutting a leaf, we can see that it is made up of cells. How we came to that level? With the invention of compound microscope. See, in your chapter, that itself starting with while examining a thin slice of cork. From there it is starting. Okay, so that scientist, he also invented this while examining a thin slice of cork. Okay, he saw many compartment like structures or honeycomb like structure which is having many little compartments. So from there the story starts. Okay, so this birth of cell biology, that is marked by the invention of compound microscope. Okay, then the next important point which we need to mention here, next important milestone, that was the discovery of cell. Discovery of cell. This discovery of cell, who did that? Robert Hooke. Robert Hooke. Robert Hooke discovered cell in 1665. So in 1665, Robert Hooke discovered cell. Okay. So this made a huge change in the concept of biology. Okay. So from the cellular level, we started learning about each and everything. So the living things, they are made up of cells and cell components. So these cells, inside cell components are available. So these cells are the starting point. Okay. So here we can say, like the cellular level, the important milestone, the cellular organization of Livingstone. Next, they discovered cell. Next goes to cellular organization of living beings. Cellular organization of living beings. Okay, so this also recognized widely, this also got recognized. So here in main points which you need to study, that is, while examining a thin slice of cork, Robert Hooke saw there a honeycomb like structure which consists of many little compartments. Okay, so cork, what is that cork? Cork comes from bark of a tree. Okay, so that comes from bark of a tree. So this happened in 1665 when Robert Hooke made this observation through a self-designed microscope. Self-designed microscope. There was a cell, with the help of his self-designed microscope, he discovered the cell or compartment like structure. Then Robert Hooke called these boxes as cells. He saw many honeycomb like structure or compartments, and he called this compartments as cell. Okay, so he called that as cell. So in Latin, the cell is the Latin word. Okay, this is a Latin word. And in Latin, what do you mean by cell? That is called, or the meaning of that word is a little room. Little room. Okay, that means a little room. Because this cell that seems to be very small and this became the important discovery. Later, this became very important discovery, the discovery of cell. Okay, so this was the first time that someone observed that living things appear to consist of separate units. Okay, after that, living organisms, non-living organisms, like that one concept. And inside living organism, there are many living units. So this living organisms are composed of many living units. That was the starting point of the discovery of cell. Okay, so first time they observed that living things consist of separate living units. Okay, so the word cell, which means a little room, that describes these living units. And now the cell, the concept of cell, that has become very important. From the cellular level, all studies are starting, right? So this is the introductory part which we can give now. So here, next concept, what are living organisms made up of? Okay, here we are going to start with the activity which is there in your textbook. So activity number 1, here we are going to take a small piece from an onion bulb. Okay, from an onion bulb, we are going to take a small piece, and with the help of a pair of forceps, we can peel off the skin, which is called as epidermis. See, while studying about the anatomy in higher classes, you will be learning about the anatomy and all. So that time you will come to know epidermis, which is the outer layer. See, from the inner side, while cutting the onion, you can easily see that inner side, outer side will be smooth, and inner side, there you can see a white layer, like a plastic sheet, like an inner layer will be there. So from the concave side, so from the inner side of that onion bulb piece, there you can remove or you can peel the skin. Okay, then this layer can be put immediately in a watch glass which is containing water. If you are keeping that peeled skin out, it will dry, right? So you need to put that in water. Okay, while doing in a lab, you will get a watch glass which is having water, and inside that you should peel this skin from the concave side or from the inner layer of onion bulb, and put that immediately into water which is present in the watch glass. Okay, so why we are putting that in water? Because it's a very thin or a very small piece of onion skin, and it's very thin, it will dry. So to avoid that condition, what we will do, we'll put that in water. Okay, next step. So first step, you need to cut a small piece of onion from an onion bulb. Then from the concave side, from the inner side, you need to peel off the skin. Then after peeling that skin, put that in water, which is there in the watch glass. In watch glass, keep some water and put the peeled skin into that water to prevent drying. Okay, so if you're putting in water, it will prevent drying. Then next step, why you need this peeled skin? Just to check what will be the, if you're watching under microscope, what you can see. So for that, you need a glass slide. One glass slide you need to take. On that also put a drop of water. Then that peeled skin of onion that you need to place on that water droplet. Okay, water droplet of that slide, glass slide. Okay, so you need to take the glass slide, then put a drop of water, then put the peeled skin from the watch glass to that water droplet of glass slide. Okay, but while placing that peeled skin of onion, you need to make sure that it is not folded or it is perfectly flat. You kept it perfectly flat on that glass slide. You need to make sure that. Okay, so while transferring with the help of a brush, in the sense the paint brush, small camel hair paint brush will be there. No, with that you can keep it on the glass slide. So you need to make sure while keeping that you need to make sure that it's not at all folded, it is perfectly straight. Okay, then after that what you need to do, next step is put a drop of saffronin solution on this piece followed by a cover slip. So first saffronin, what is that saffronin? That's a dye, okay, which is helping to color. So that dye you need to put saffronin, you need to, that's a coloring solution that you need to put one drop, you need to put on this. Then you need to put a cover slip. So cover slip, when you are putting a thin cover slip, that's a very thin, just like a slip that you need to put on this peeled skin which is present in the colored solution and water. So if you're putting, take care that no air bubbles are present inside. So that if you're putting a cover slip, that will make the, that will help for mounting or mounting you with the help of a small needle. This and a lab in laboratory you can see while doing this activity in class, it will be very clear for you. So what you can do, first you need to take a brush, with the help of that brush you can take that peeled skin from the watch glass and you can place it on this glass slide and put a drop of water, then saffronin, which is a coloring agent or which is a dye that you can put one drop, that gives color. Okay, then you need to place the cover slip with the help of a needle. And while placing that cover slip, make sure that it's kept properly without any air bubbles, and the peeled skin, that also make sure that it's not at all folded. Okay, so you can, with the help of a needle, you can mount the cover slip. Okay, so now what we are doing in all these steps, we are preparing a temporary mount of onion peel. It's a temporary slide or mount of onion peel we are preparing now. Okay, that's what we are doing in all these steps. So we can observe this slide under low power. First, you need to watch the mic in a microscope under low power. Okay, magnification should be less first. Maybe 10x or 5x you can watch this. So that is in low power you can watch first. Then you can just now how we are doing that zooming, same like that you can go for next level or high power of a compound microscope. Next you can watch. First you can watch under low power. Next you can watch under high power. Okay, so here this is how, here you are preparing, this is the glass slide and here you are placing your onion peel. Onion peel with the dye and you're mounting with a cover slip. Okay, then after placing, this is a microscope and here you are keeping the slide. Then what you need to put, microscope slide. This is a slide. This slides will help to keep the here you can keep this is a slide, you can see here, this slide, and here clips are there. These clips will help to keep the slide in a position. Okay, it will not move. Okay, so while going to the high power also, you can was the same place. That's why they are giving that clip to protect the slide without changing the position. So these are the objective lenses. Here one you can see, and here also one you can see. One will be having lower magnification and one will be having higher magnification. Okay, so here what you will do, this is the mirror. Okay, so on this mirror light will come. So this is a condenser. So through this light you will get light towards upward, and this is the eyepiece. You will be watching from the top. From here you can see, and with the help of this objective lenses you can watch what is present. That onion peel or that peeled skin of onion you can watch here. Okay, and this is the fine adjustment and this is the coarse adjustment. With this you can see, you can adjust the length where you can see the structure without any disturbance. See, you can see structures like this when you're watching from eyepiece. So again, fine adjustment you are doing, means you are making clarity in fine adjustment. Okay, so then the objective lenses, we said one is having lower magnification, one is having higher magnification. So first you will be seeing like this in lower magnification. When you are going to higher magnification, only this one portion you can zoom. Okay, so you can see a fine structure of that. Okay, that's how you're going to do. So we can observe the slide under lower power, then you can observe under higher power. Okay, so while looking through that, what do we observe when we look through that lens? What we can see when we are looking through that lens? So while observing through the lens, first we can draw the structures. See, this you need to draw the structures like this. Okay, it's very easy, very simple experiment. You can see through the microscope and you can record all the observations in an observation sheet. So you will see a structure like this. Okay. These structures, these are called as, see, each structure you can see in this. These structures are called as cells. So this is a cell, this is a cell, this is a cell. So you can see many cells together, and you can next under this higher magnification lens, you can zoom one portion and you can watch a single cell. So you'll get little more clarity. So while watching under a higher magnification, what and all you can see this structure. See, now I'll mark one compartment of that. See, this is a cell, right? Okay, like this you can see a cell. And inside cell, you can see a dot-like structure which is called as nucleus. Okay, here you can see a nucleus. This is the nucleus. Okay, next, inside that you can see vacuole, a vacuole. Clear? Then the outer one, this is the cell wall. Here you can see the thick portion, the outer wall, which is the cell wall. Then cell membrane. Here, see, when we are using that color solution, that epidermis or the layer, the compartments that will get colored so that we can visualize or we can make out each and every cell separately. Okay, the cell wall, the outer layer, that will get colored so that you can see each and every cell separately. This is due to that dyeing or coloring solution saffron. Okay, that's why we are coloring that, to see it separately. Okay, so this is called as cell. With the help of a compound microscope, you can clearly watch a cell. And here you know that living organisms are formed of cells. They are made up of cells. All living organisms are made up of cells, and these cells, that is the structural and functional unit. These are the structural and functional unit of life. So living organisms are made up of cells, and these cells are the structural and functional unit of life. So this cell can be regarded as basic organizational unit of life, basic organizational unit of life. And one more thing, which is the very important point, this cell that is capable of independent existence. This can exist independently. Okay, and this can multiply. These both points are very important. See, cell, this can be called as the basic organizational unit of life. Cell is the basic unit of life. Okay, and they can exist independently. Independent existence, that is possible for this. Which one? For this cell. And that can multiply. These cells can self-multiply. And sometimes some changes may happen. These changes are called as mutation. So some changes may happen, which is called as mutation to this cells. So what we said, this is the basic unit that can multiply and they can undergo changes. Then they are having the power. They are storing that their character or the molecular character they are storing, and they have the power to transfer, or cells can transform, they can change, and they can utilize energy. They can maintain the environment, proper environment, cellular environment they can maintain, and they can undergo duplication. So cells, they are independent. They are independent unit. They are the basic organizational unit of life, and they can exist independently. They can multiply, then they can store all information. They can transfer that information from one to another. Then they can transform, they can change, and they can utilize the energy. They can maintain the cellular environment. So a cell is having the capacity to exist independently. Okay, that's what we are saying. That is the conclusion of all these points. These cells are having, each and every cell is having the capacity to exist independently. independent and basic unit of life, that is cell. Okay, the cell is the basic unit of life. It is the independent unit of life, and that is having the capacity to exist independently, that is having all informations, and that can store the informations, that can pass the informations, and they can change the information, that is having the capacity to change the information. Sometimes they'll undergo changes, which is called as mutation. Then they can maintain the cellular environment. Okay. So organisms, they are of different types. Organisms may be unicellular. Unicellular means with single cell, and sometimes multicellular. Unicellular organisms and multicellular, more than one cell. Multicellular organisms are also there. Unicellular means they have single cell which can do all these processes, whatever I said. It is having a single independent unit, or a single cell which is working as an independent unit. But in the case of multicellular organism, if you are separating multicellular, suppose it is having 100 cells, that organism is having 100 cells. If you are separating each and every cell, it cannot exist. But it is having all the informations or all the things which are there in a single cell, but they are jointly working. They are having a coordinator or mutual cooperative work. So when we are separating all these 100 cells, it cannot exist, because they are working in a cooperative way. Okay, with a mutual cooperation they are working, and they are not independent. Okay, so when we are keeping all these together, all these 100 cells together, they'll work in a cooperative way and they will perform the functions. But at the same time here, when we are separating the cell, that organism cannot exist. But if you are keeping that together, that cooperative way, that organism will work and that can exist. Okay. So how we can explain here cell of unicellular and multicellular organism? See, cell of unicellular organism, unicellular organism means an organism which is having single cell. Unicellular organism can carry out the whole life activities. That can carry out whole life activities. Okay, so unicellular organism can do all the life activities by itself. Okay, and with the cell and cell organelles inside the cell components are there. So with the help of all these cellular components, that unicellular organism can carry out the whole life activities. But here in a multicellular organism means organism which is having more than one cell. So in multicellular organism, here you can see an individual cell. Here we cannot separate multicellular organisms cells, and we cannot ask them to exist independently. Why? Individual cell of a multicellular organism, each and every cell or individual cell of multicellular organism. Each and every cell of a multicellular organism is specialized for a specific function. Specialized for specific function. Okay, so here multicellular organism will be having more than one cell in their system. So in that multicellular organism, if you are separating each and every cell, those cells are performing different functions. Each and every cell will be performing different functions, and these functions together makes that in a cooperative way all these cells are performing different functions, and that makes a multicellular organism. Okay, so if we are separating, we can see each and every cell or a group of cell that will be performing a specific function. So there is a clear division of labor. Division of labor. Clear division of labor among the cells in a multicellular organism. And these cells, they will cooperate. They'll cooperate with each other to form a coordinated group for a common function. So in multicellular organism, cell has dual existence. Okay, like it is existing as a single individual and as a member of community or as a member of group. That cell can individually exist, but to perform a function that will work as a member of a community. And next is very important. In unicellular organism, if one cell, the cell death of a cell in a unicellular organism means death of whole organism. Unicellular organism means only one cell. There is only one cell. If that cell dies, whole organism is dying. Right? If unicellular organism is having only one cell and that cell is dying means what will happen? The whole organism will die because there is nothing else. That whole cell is dying means that organism is dead. That's all. But here in multicellular organism, death of a cell, death of a cell or few cells in multicellular organism. So in a multicellular organism, there you can see division of labor, right? So one particular group of cells will be performing one particular function. If those cells in that one cell is dying or that group of cell is dying means that particular function is getting inhibited, right? So division of cells in unicellular or death of cells in unicellular organism means that whole organism is dying. But in multicellular organism, death of a cell or death of few cells, that will not affect that organism in a serious way. Why? Because their one function is getting disabled or one function is getting laid, or those cells are dying, or the one cell is dying or a group of cell is dying means it is meant for a particular function. Division of labor is there in multicellular organism, right? One group of cells will be performing one function, so that function will get disabled. That's all. It is not going to affect that organism in a whole way. The whole organism is not going to die because other cells are there, other group of cells are there, they'll be performing their function, right? Then next point, so first point, unicellular organism means all activities are performing by that single organism, and death means that whole organism will die. But in multicellular organism, each group of cells will be performing each function, and death of a cell or death of a group of cell means that doesn't have a meaning that that organism is going to die, because remaining cells or group of cells will be there by performing their function, right? Next point which we are going to tell is division of cells. Unicellular organism. In unicellular organism, division of cells. Division of cells. That means asexual reproduction. Asexual reproduction. Okay, but in multicellular organism, see here in unicellular, don't get confused. this is a single cell organism, this single cell with nucleus. This organism here, division, this is getting divided. Okay, so this is asexual reproduction, reproduction without any sexual process, that is getting divided into two new organisms, that's all. But in multicellular organism with more than one cell, that means again cell division means that organism is growing. Again, one cell is here, several cells are there. Okay, so in such organism, cell division, this cell is dividing and this is here number of cells are increasing, means cell growth. Then one cell is dying here, this cell again division, it's for cell growth, cell repair, then regeneration. Okay, that organism is dividing, the cells are dividing in that organism and it is growing, growth is happening, repair is happening, some cells are getting damaged. So again with cell division, that repairing work is done. Okay, so that's what we mean by the cells of unicellular and multicellular organisms. Cell is the basic unit of life. And what do you mean by cell of unicellular and multicellular organism? Cell of unicellular organism can carry out whole life activities by itself. Then individual cell of a multicellular organism. In multicellular organism, each group of cells are meant for specific functions. So that there is a clear division of labor among cells. Okay, so multicellular organism, that cell is working as an individual cell and it is existing as a part of a community or a group of cell. Then in single cell or unicellular organism, uni means single, the single cell organism, there a cell is getting damaged or dying means the whole organism is dying. But in multicellular organism, death of a cell or a few cells or a group of cells are dying means it will not affect that organism seriously, because division of cells, there is a division of labor, division of cells. Okay, so other cells will be performing their function, so it will not affect that much seriously. Okay, then in unicellular organism, division of cells, cell division means that single cell is getting divided and it is forming two new cells. So that is asexual reproduction, that means without any sexual process, that means asexual, a reproduction is taking place. Okay, but in multicellular organism, cell division, a division means the number of cells are increasing in that organism, that may be for growth, repair or regeneration, for different purposes. Clear? So here that much only we are learning about general concepts of cell. Then few more points are there about cells and different types of cells that we'll learn in the next class. Okay, thank you.